This is just the name that I prefer to put this, drought tolerance in sugarcane. This is a preliminary study to look into the water economy of these varieties that Marty and Eric sent us. Actually, all of these started when I was vacationing in California and Oregon and Colorado. So my research associate, Carlos Correa, had to take, and Dr. Landivar had to take hands on this and get it running. So when I came here, everything was already planted and going. So I'm very appreciative of that. This is the Drought Tolerance Lab that we've been using to test these canes. Originally, this actually this is these two old greenhouses were doomed to be destroyed, but thanks to Dr. Landivar and in a conversation, we saved the structure and we replaced it with a high transmittance roof, removed the sides, so we have free airflow, and there is no need to do any air conditioning control or anything. And it's very close to the outside temperatures. Originally was developed for cotton, to do research on cotton, because we had a great interest. That year we had a Monsanto test in the at the center where they were testing different varieties, and we could see the difference in there. So this idea came up of saying, well, let's build this drought tolerance lab in order to start not only characterizing the water economy of genotypes, but also identifying traits that control the water economy. And the idea was to integrate different levels, the agronomists or physiologists like me, plant breeder, plant geneticist, and all of us benefit from understanding what is controlling the differences in water economy among these different cultivars. So we ran 3 years of this research in cotton, and at the end of the third year is when Dr. Landivar proposed to do some, to look into sugarcane varieties, especially the ones that were developed that were showing or exhibiting these drought, cold, high temp resistances, tolerances. Anyways, so we set up the experimental method is the phase 1, which is a well water growing conditions. We didn't restrict the supply of nutrient solution to these plants, and we had these 16 sugarcane transgenic varieties. So we had 3 plants of each and 4 reps, and these are the materials that Dr. Dickman and Mirkov provided us. So we had a total of 192 plants, and 64 of those were hanged and monitored continuously for water use, daily water use, or actually every 10 minutes, using Wagon mini lysimeters. You're going to see some pictures. So they were planted in relatively large pots, 13.5 liters, with exactly the equal volume of dry fritted clay soil, which is the material, the medium that I like to use because it has a very high water absorbance capacity compared to the other potting materials, and is very stable. And with that, we remove part of the variability for differences in soil. These were planted on September 27, and the emergence occurred on October the 5th. They were irrigated in excess till producing drainage using an automated system that supplies the nutrient solution, and we were irrigating every 24 hours at 24-hour intervals. And we always provide the irrigation at night, and I'm going to explain why in a minute. So we monitor, these plants that were monitored were harvested for dry mass distribution after about 20-something days, and leaf area measurements on December 18. At the same time, we've been measuring the weather conditions inside the, it's not a greenhouse, actually it's a rain shelter, temperature, relative humidity, radiation, wind, and along with the lysimetric data. Everything is automated, and these lysimetric data and the weather were recorded every 10-minute intervals. This is a shot showing an early stage of growth of these canes, and on the right, the shot shows at the end of the second phase after we expose plants to water stress for a long period. And when we saw these green plants there, we got pretty excited. But as we analyze the data, the excitement kind of disappears a little bit, so we're going to be discussing that. It's still a good thing to see that plants remain green after long periods of drought. The daily plant water use calculations are, as I said, daily. We summarize the data on a 24-hour basis, and it is just the sum of the differences in pot weight between consecutive hours. The reason for doing this is that the amount of growth that accumulates in 1 hour is much less than the water exchange or the water loss in the plant, so it's a pretty good way to just compile water use. We take the records every 10 minutes using the Wagen lysimeter that produce millivolt output, and this information is uploaded to a server for processing using a web-based program that I developed. It's not published. This takes all the data and does a lot of processing, intensive processing, summarizing the data. So basically what it is it gets 10 minutes output voltages, it converts that into hourly pot weights after doing some cleaning that I'm going to show in a little bit, and then from the hourly pot weights, using the calibration, we get the hourly plant water use, and that is summarized into daily plant water use. Calculation files and then exported to Microsoft Excel for processing, further processing and graphical display. On top of this graphic shows is a sample of a 10-minute interval load cell outputs. This graph on top is made up of what 2,857 data points there. And those spikes that you see, these spikes are the irrigations that occur during, and this is only one plant. I'm showing this as an example. We irrigate in excess. The soil medium is very permeable, so as soon as we add the quantity, obviously the load cells see that amount of water going into the pot, but it drops immediately because of the drainage. So all this, and what we see, this progression, let me show you here. This is the irrigation at night, then the water loss that occurs at night, because it does occur a little bit of the, loss. I mean, it's not completely zero. During the daytime, I don't know if you see the map. Do you see? Yeah. Do you see the arrow there? This is the night loss, and then the daytime loss, and these ups and downs, and because clouds passing or some condition at the end of the day that finishes, and then another irrigation and so on. So those high values need to be removed, and the program that I wrote does look into the data and remove those spikes in order to do the calculations. And the bottom graph shows the data after the removal of those spikes. This is a sample of data consistency. What is important here to see is the parallelism that there is in the lines. These are 8 different plants there, and through 19 days, and we see this parallelism there. And the spread is caused by differences in plant sizes, mainly. Basically, with all these stories says, well, the method works. We can really measure good things there, and it provides good information. What I'm going to do now, next, is I'm going to run the graphics and I'm going to explain what is in these 3 things here for the 16 varieties under the well water conditions. And the red line represents the average of all the entries, of all the varieties used, and the blue line represents a specific, the specific entry or genotype. In this particular case is 11. We see that the whole plant water use, or the daily transpiration per plants in milliliters per day, is lower than the average for 11, and then the question immediately arises is, well, why? I mean, is this plant size? Is it transpiring less, or what? We see in the bottom graph, column graph, that this plant 11 has a much smaller leaf area, total leaf area, than the average, the average of the genotypes. So then we can start suspecting that that behavior that we see in the daily transpiration is caused because is such because of the smaller plant size. On the right hand, we took the daily transpiration and we divide that into the per unit leaf, well, by the leaf area in order to get the transpiration per unit leaf area, which is an important trait, an important characteristic for the plant. And in this particular case, it shows that the daily transpiration per unit leaf area for this variety is really much higher than the average. So this is what I would say a prodigal variety. The reason that it transpired less or used less water than the average is because they were small plants. This other variety, this is 15, shows also that the daily plant transpiration is less than the average. Now, if we look at the total plant leaf area, we see that the leaf area of this plant is pretty close to the average. And when we do meaning that then it has to be the transpiration per unit leaf area. In this particular case, it shows that the transpiration per unit leaf area of this variety is lower than the average. So this is a water conservative genotype, okay, which has an average leaf area. And we can go so on. I don't know if it is worth to go explaining in very detail all of this. This plant, 20, is very prodigal. It uses a lot of water. The plants are bigger than the average, or the leaf area is bigger than the average, and also the leaf transpiration per unit leaf area. So this is, if we are looking into water conservation, this is not a variety that I want to use. And we can go on. So we have been recording all this data, and we've been presenting this for all the varieties. So I'm going to flip over relatively quickly. This is also a water conservative genotype, but with a larger leaf area. Okay, the reason that the daily transpiration per plant is close to the average is because of the bigger plant size, but it helped that the plant transpired less per unit leaf area. This might be a good one because if leaf area is related to biomass, then we want to have a water conservative plant that produces biomass. 69, this is another variety. Let's go over 112. 112 is also, it caught my eye at the very beginning. It has a much lower daily plant water use, but the plants are smaller. And the transpiration per unit leaf area is just the average. This one has a very, to me, it has a good rating in the sense that it uses less water, it transpires less per unit leaf area, and it has close to the average leaf area. This is also a prodigal water use plant genotype that uses a lot per unit leaf area. This is probably not one that I like to use either. And so on. So we can go flipping these materials, these varieties, and so you can have a grasp of the kind of data that we can collect in this facility. 170, 171 is one that is giving you a good water conservation, also with an average leaf area. Obviously, we also calculated the water use efficiency, although the value that we have is not the true water use efficiency. We just divided the total biomass that we have for that plant and the amount of water that used during those 20 days that we've been monitoring each of these plants. So actually what it is a much bigger dry weight per a smaller amount of water used, so the values are going to be higher than actually they are. This is the variety that sort of, when we had the first run and we had a few plants running, we found that this plant has survived alone without many days without water. And it shows here that this behavior was good. I mean, it used less water, it's close to average, and so on, but there are other better individuals. There are genotypes that we found. This is another one, and this is the last one that we have. 1210. Yeah, it's a bigger plant, and it has shows that under well water condition, it has a lower water use per unit leaf area. We run a statistical test of this. One of the things that I want to show and point out very clearly is the high coefficients of variations that we have here, 26, 31, 47 for the total value of water loss, biomass, and water use efficiency, which is the ratio of the two. This analysis show that the variety 15 that I've been pointing out, and 158 and 170, 133, have pretty good ratings in terms of water use efficiency, but these values are really not realistic, okay? They are kind of inflated. These other varieties, 1210, which is the one that you were asking, and 33, 38, and 20, and 69, have lower water use efficiency. So if you look at these values in here in general, let's take 15, for example, that has the highest water use efficiency. It has a high production of biomass. We are not talking about leaf area here. We are talking about biomass, and below average water use, which is a good combination. Biomass production, low water use. That's why it gives a high water use efficiency. Then we enter into the second phase. I said, okay, let's get serious here. We are going to expose these plants to water stress, to water deficits, and to create water stress. The plants that we used in the first phase and the well water conditions were harvested at the end, so we took the extra plants that we had that were just on the side of the other one, and we put it in the lysimeter, and we start measuring the water conditions. The plants are not exactly the same because there is variation in plant size, so even though there are three plants of the same genotype, they are not going to be exactly the same. There are going to be differences in plant size. That's important to keep in mind. So what we did is we decreased the irrigation to 25% of the well water level by cutting in half the amount of water that we were applying, but every other day, instead of daily, every other day, which makes 25% of the maximum watering. So the first stage of 25% caused a moderate water stress, And later, a few days later, we completely suppressed the irrigation on December the 27th to initiate a phase of severe water stress. This is just a repeat. The 65 plants were continuously monitored, and then at the end, these plants were harvested. Now, because we were seeing that some of the plants were still green and the others were pretty, with the foliage pretty dry, senesced, we said, well, we need to have means, we need to do something in order to capture this. I took photos, but we need numbers. So what we did is we used the main column or the main tiller of the plant that was already tilled in a lot. And these plants are bigger than the previous ones because they were planted at the same time. And we harvested all the leaves in the main column, the leaf lamina, not the sheath, but the leaf lamina, and hoping to capture and measure the fresh weight. Immediately after cutting, all the plant parts were measured for fresh weight. So the idea was to capture that more hydration in those leaves, those greener leaves. The ones that were very much senesced would have a much smaller water content than the ones that were still green. And let's start looking at some of these. The presentation, the data shows a little different than the previous set. This shows the graph on top. Red is always the average, and blue is a specific variety or genotype. And the arrows indicate when the irrigation was put down to 25% and then the no irrigation. It shows very clearly that even at 25% irrigation, those plants continue to grow a little bit. I mean, it was just light stress, moderate stress. That's what plants do. They still grow and survive. The growth rate may be a little limited, but they still grow. And something that I forgot to mention, on the ups and downs in the lines are the weather variations, induced by weather variations. Okay, when sunlight, the amount of radiation, the amount of wind, and the temperature, the humidity, all these factors are controlling that. That's why those lines go up and down. When we put the plants under severe stress on the 27th there, then yes, I mean, they really didn't like it, and they start reducing the amount of water use. The graphs, the column bars on the right-hand charts, the top one shows the water content in the main tiller. I mean, this is the fresh weight of those leaves. It's not relative water content, it's the total amount of water that we found in those leaves. And this value is going to depend not only on the water content of those leaves, but also on the amount of those leaves. On purpose, I didn't want to show the relative value, okay? Because then we have the bottom graphic shows the total leaf dry mass, which gives us an idea whether the amount of water that we found there is somehow related to the amount of leaves, obviously. In this particular case, 11 shows that there is a lot of water in there, much more than the average, even though the leaf dry mass is below than the average, indicating that those leaves were pretty green. And look at the photograph below, it shows a lot of greening in those leaves. These are actual photos of these pots, of these plants. And, well, we did the same analysis in all of them. In this particular case, 15, you see 15 showed to be a pretty good genotype under the well-water conditions. It's using a little bit more water in here. Remember, plants are a little different, but also it shows that the foliage is pretty dry. Well, this is average. If you look at the graphics here on top, this is about the same as the average, and the total leaf mass again the same. But this is the one variety or genotype that was plotted as being good under well water conditions. 20 was not that good. I mean, it's using a lot of water. The same thing with the other ones. The foliages are pretty dry, below the average. And these ones, a lot of foliage too, more than the average, and the water content in the tillers about average. And the water use, the transpiration per plant is of these genotypes on the average is pretty similar to average. Again, 69 wasn't a good variety or genotype in the previous one. It uses, but it produces quite a bit of dry mass. Daily transpiration, this is one of the varieties that when we saw, we got very excited. This one, and I think it was 11 too, and but we are finding now, if you look at the water content of those leaves, is good, quite higher than the average, but the total leaf dry mass is lower than the average. So to me, this is telling me this is a good graphic to see, but the plants are small, which is not, might be the reason why this variety is you know, remaining pretty green. A smaller plant that is using less water. 132, let's go flipping on this. 133, it shows that it desiccates, although there is one of the reps that shows pretty green there, but probably very thin, small leaves. 136, 158 is one of the genotypes that it behaves relatively well. It uses less water during the stress phase. The water content is about average, but it produces less than total dry mass, leaf dry mass than the average. 163 didn't caught my eye. This one so-so, and 171. It shows good water content. I mean, lots of green leaves in there, despite the fact that the plant it has a lot of leaf dry mass, more than the average, and the water loss is similar to average. So to me, this is like a good candidate to look at. 375 again produced a lot of biomass in leaves, but the water content is not that good because also the total leaf dry mass includes everything, okay, and the water content is only on the main tiller, so we have to be aware of that. There is the amount of tillering in the plant is different. 110. Just very average, and the other one, 3388, uses a little bit more water there, and is a bigger plant too. These are the summary of the statistical analysis we did there, where it shows on the right hand the water use efficiency, which is the value that I want to be looking at, is 158 and 171. Pretty different than the ones at the bottom, 11, 133, 163, and so on. This combined the fact 158, for example, if we look at this, is a medium amount of biomass in there, but it uses very little, it's in the bottom of the list there in terms of water use, so it's water conservative. So the conclusions that I have, the method is good, okay? It allows a very accurate monitoring of the water use patterns among individual plants. And I'm not, before I had the word genotype there, but I changed it this morning, very early in the morning, like 4 in the morning, to individual plants, for one reason, which is explained later down here at the bottom of this list. the method is good to follow and tracking, monitoring water use. The fact that we put the same amount of soil, we eliminate soil as a factor, we have very good controls, all the mechanics and all the automation and the irrigation is good, but, and we are able to pick up differences in the water economies and either both well water conditions and water deficit. Plant size, either leaf mass or leaf area per plant, and the transpiration per unit leaf area are important plant water use controlling factors, and we were able to see there. A small number of genotypes were identified as water conservative and water use efficient, like 15, 171, 158. Okay, but there are still some unexplained inconsistencies when we look at both water regimes. And we were looking just at the water use efficiency, at the biomass and leaf area and so on. But the main point that I want to make on this is the bottom paragraph, the bottom bullet. The technique is good, but one of the things that we observed in this preliminary study is that there was a large variation in plant size. Okay? Remember, we are coming from doing cotton. In cotton, we select the seeds, we seed the seeds, we have seeds of the same size, we have tremendous amount of uniformity in the plants that come emerging. Okay? And even 1 day or 2 day difference emergence, or 3 day difference emergence will create a big difference between those plants that have different ages in emergence. So, in this case, although I didn't see the propagates, I didn't see the seed because I was vacationing, I can imagine, and Carlos corroborated, we talked about this a few days ago, and Carlos corroborated this, the amount of chunks with the meristems to grow, to develop the plants. The size of those chunks were different, the thickness was different, some thinner, some thicker, different length, and so on, and that is the source of substrate that the meristem has to grow, for growing. And that's an important factor because it determines the initial growth rate of that plant. So, in order to continue with this and being more accurate, and then start talking about, oh, the method is good also for monitoring or to detect differences in water use among genotypes, I want to see a more uniform propagate that we plant in those pots. That will reduce a lot of the error in all of this, and it will improve the statistical differences among genotypes. If they are very different, like the case that we saw in this preliminary study, that compromises the statistical analysis because of the noise in the data. And that's all I have to say. So if you have any question, I want to acknowledge and thank Carlos Correa. He is the person who made this run, and also Raimundo Gonzalez, who is a student with an intern student from Del Mar College that has been working hard also to measuring biomasses and leaf areas. And Kenneth Schaefer, yes, Kenneth Schaefer it's a very important contribution here too.